Hi.
Okay, now we're recording. I'll start that over again. Welcome, everyone, to the first of hopefully many Asian brown bear expert team webinars. We'll be recording, and if you could turn your audio off so it doesn't interfere in any background noise. And Dave's going to share it out with a wider BSG. I think that these webinars will probably be pretty interesting for a lot of people. Well, tonight we have Odka Tumendemberal. I can say her name really well, but I got tricked up. She's just a recent, well, in the last year or two, got a PhD out of Norway with Andreas Zedrosser, and has done a really extremely good job of publishing all her PhDs. She's just a kind of a rising star in Asia. And I met Odka in 2008 when I got dragged over to Mongolia to work in the Gobi Desert with Harry Reynolds. And Odka was just fresh out of undergrad university, and she wanted to be a geneticist. You know, she was this quiet, young little thing, and they made her my translator so we get to know each other. And sure enough, within a couple of years, I could see the underlying drive of the Odka. She's a serious, hardcore, hard worker. At any rate, her talk tonight is going to be, hopefully, we'll see what it's really about. But what I asked for was to give a summary of all the trend analysis that she's done over the years in multiple DNA surveys in the Gobi Desert, this very isolated population. And then she's going to talk a little bit more about some of the phylogeographic and genomic information she's gleaned. She's become quite the geneticist. And at any rate, we're real happy to have you, Odka. Thanks for doing this and getting us started. And I'm going to turn it over to you so you can share your screen.
Thank you so much for this opportunity. And I'm so happy to be here and meeting you all. Okay, let me share my screen.
All right, we can see it. Perfect.
Sorry. Okay.
Here we go.
Hello, everyone. So today's presentation is on research, conservation, and future perspectives on brown bears in Mongolia. And today's talk will have following order. First, brief introduction. And second, we will talk about what we learned in this. We have, as Michael described, have these two studies results we will check, and then lastly we'll check the future directions. So brown bears are believed to be originated in Asia and distribution extended throughout northern hemisphere. And however, the historical distribution is brown bears has been decreased and it become fragmented, small, especially in southern range. And in general, in brown bears are well studied in North America and Europe. However, there's limited information in Asia, in Central Asia, and sometimes endemic population can go extinct without knowing the limiting factors. So to conserve the population, we need to know all this demographic, evolutionary history, phylogeography, genetic diversity, and all taxonomy, and all this information to have a better conservation implications. And the research consists of two main chapters. And first two work is on demographic and Gobi Desert. And we did first one have sampling from 2009. And then we continued this work over the years, and we have 3 years systematic sampling, and we also had opportunistic sampling from 1996 to 2007. And all this work, those two are published. And then second one, we sampled from brown bears in Central Asia, including 8 geographic regions, used microsatellites and then some partial mitochondrial DNA, and this paper published. After that, we had to learn more about more details of the evolutionary history and use whole genome, and we will discuss those results. First paper. So this demographic was one of the goals for Gobi Bear project, and we started in 2008 as a pilot study. As you can see in the picture, Michael is there, Harry Reynolds is there, and in this team, picture is from 2008, and we had a team including rangers from Great Gobi, where Gobi bears are distributed, the strictly protected area, and then researchers and student, and there's one photographer, and we had a good team started this work. And before we start this work, we can look at Gobi bears. And Gobi bears are considered to be a pride animal in Mongolia, even it's a legally governmental legally made it a Mongolian pride animal last year. And Gobi bears exist very unique environment, and estimated in 2005 they were like population size. We knew it's a small population, but from 20 to 5 there was a guesstimate based on observation and tracks, and the distribution reduced, especially 1950s to 70s. And Mongolian, it's listed as a critically endangered, and hunting is prohibited 1953. Once people know there are rare bears in this region, and distribution range was protected 1975. The Great Gobi established that, and feeding program, the supplemental feeding program started 1987. And we had at that time limited knowledge on movement between the mountain ranges, because in Gobi, Great Gobi has 3 main mountain ranges, and people question that there might be isolated tree population, even there were questions on that. And genetic diversity and population structure and demographic, all this information was necessary to be answered. Okay, so for study area, as previously said, the Great Gobi is strictly protected area. Is in southwestern part of Mongolia, as you can see in the picture, in 3 mountain ranges, and red dots are showing the sampling sites, 13 sampling locations. Those are main water points where we built the hair snares to collect the hair samples, and the gray area came from our special capture-recapture model showing possible distributional range. From here, this picture, you can see that how Great Gobi look like, very rugged and remote. And this is one of the larger oasis complex in Great Gobi, Sharhos. And for sample collection, you can see that barbed wire, we used feeders and trees.
Odka, if you can hear me, I think your internet froze.
I don't think she can hear you because
I don't think so. Joel, can you hear me? Odka's internet froze. because internet died. All right, well, if worst comes to worst, I'll finish that talk, but I think hopefully she'll be back here in a couple of minutes.
Michael, I have a question. She said that in 2005, an estimate of about between 20 and 50. How about now? It's 2024, after 19 years. Any estimate right now?
Yes, she'll tell you. I'm not going to tell you.
Okay. If she can return to this talk, of course.
Tell you. The cool thing is that, you know, it's pretty hard to estimate trend, you know, to watch a population change. So what they did was cool is we did a survey in 2009. And she repeated it every 4 or 5 years and had good enough data that, you know, she could really hone in on a bit of a trend.
Right. Got that. Thank you. Darem, are you? Oh, she's back. I think she's back.
Okay, you're back. Very good.
Excellent.
Oh, you're on mute again.
Sorry, I changed my room. Hopefully now it should work better. Fine.
Share your screen.
I'm sorry for this.
No problem.
Okay, I'm going to share my screen. Okay, you can see the study area, the sample collection.
Yeah.
No, we can't see that. We
I can't
can't
see the screen yet.
Oh, yes, sorry. I need to share my screen first.
Yes. All right, there we go. Now you can start your talk again.
Okay.
There you go.
Okay, so 2008, we first established this barbed wires, Megalcom, and we built this first hair snaring sites, and we used also trees, larger trees nearby water, and bears like to wrap their backs, and we also collect the samples from there. And you can see the good sample from this picture. And from 2009, 2013, 17, we had systematic sampling. And for these years, we were able to, using those repeated sampling, we are able to estimate population size, survival, and density estimate. And as previously said, we have opportunistic samples. Those samples are from those years during sampling time, also bear tracking. And when we have different years of those systematic sampling, and also when we find samples outside of the springs, some scat samples they will be included for keep track of individuals for the monitoring. And here our field work, sampling worked, and we were very happy the first year. You can see a picture of. For the laboratory analysis, we extracted DNA and identified individuals using 13 polymorphic macrosatellite loci. We initially started 24 and most polymorphic 13 macrosatellite loci for Gobi bears. And we used, we also did a species ID first and a macrosatellite loci, and also identified 6 ID. And as you can see from the picture, I first got the training in Canada, Nelson, BC, and second, since 2013, I've been worked in Lizard Waits lab in University of Idaho. Here you can see the picture, and we were able to identify individuals over the years. And we first data analysis we used based on this repeated sampling, genotype information, pairs information. We did capture mark recapture model, and first year data we used Huggins closed model and heterogeneity. We used time and 6 as the coverage, and for second paper now we have 3 years, and when we have more year data we were able to use more stronger. The data makes it more statistical analysis make more stronger the results show, and we were able to use robust design. And here we can use both open and closed model together. And for year 2009, 13, 17 are closed to estimate population size. From 2009 to 13, 13 to 17, those years, over time interval, we are able to estimate appearance survival. And we also were able to use spatial capture recapture, multi-session secure model, and based on that, individuals are moving between houses complexes, and based on that information, we are able to use this model. And we estimated the population size, and we used null and full models with year 6 and its interaction with detection probability and activity center sigma. And we also estimated annual population growth, and AIC was used to select best supported model for both of those analyses. And here you can see the number of samples we analyzed over the years, and you can see a number of successful genotypes there. Totally for systematic sampling, 990, almost 1,000 samples. For opportunistic samples, 26, and we found unique individuals. We can see the number, it's very small number, different individuals. Over 3 years systematic, we found 51 unique individuals, but it doesn't mean we have now 51 individuals. There are a wrong number going on there. And for opportunistic samples, we found 40 new individuals, totally over time 65 individuals. And looking at those results, the sex ratio is skewed towards the males, and mostly like 1 male, 3 males, 1 female ratio to 1. And here, based on the genetic monitoring, we found 65 different individuals over the years, and each column representing unique individuals, from the blue showing the first detection to the last detection. We can see how individuals changing the over time, and each time of sampling, we found new individuals and also found some individuals as previously we find them. And the green ones are the captured bears, they put colored, and we also had sample. And we are based on those results, we were also able to see how individuals moving between the houses, how many numbers are detected. It's the results from 2009, and female movement are less than males. You can see there are your individuals are moving between those houses complexes, and also males in 2009 here, more number of males. 2013, there are also this number of males moved between this area. Now let's look at the results. For mark recapture analysis, the top method was the survival intercept, affected by anything, and then detection probability was varied by mixture covariate, and detection probability, 19% of population have high detection probability, like each session, and 81% of population have 0.3, 33, and it's pretty high. It's each time of a session we find this percentage of population, and we were able to detect most of your individuals in this population. And for survival rate, it was 0.85. For the secure model, the top model was density wasn't affected by any covariate, and for detection, baseline encounter probability was influenced by 6 and year, and sigma, it's kind of home run, similar, changed by year, and mean maximum distance moved was 47 kilometers, and mean annual captures for each individual 12 times, mean special capture each individual in one location 2.5 times, steady space each 1,000 kilometer, there's a one individual there. And now let's look at the discussion on the results. For survival rate and annual population growth were in our survival rate similar to other brown bears, and mean annual population growth was little over 1, so it appears to be stable. But we know that Gobi bears are not hunted. Comparing rest of brown bear, other demographic studies, a lot of populations. And for why there's a skewed sex ratio. So it can be, when our initial guess was it can be sampling bias, because we know that population should have 50-50 males and females 50%, but and we were guessing our first estimate they were more males, and we thought maybe males are dominating and females may avoid coming to this sampling area, may not give in the samples. And we built additional sampling points. Each of these sampling sites have another additional in one nearby area built supplemental food area, and then built a sampling location in, but they didn't help. That helped you capture more females, but this didn't help. Still, it was same result. And it's possible that through sex ratio, and but both method, no method were didn't show any biases on population survival and density estimates. And we also, it's possible that the females may be taking extra energy for raising their young, the offsprings, and have more stress, and they may have a lower survival rate. And when we looked at previous mortality causes, the records, there were more females and cubs in that list. And further, so Gobi bears are vulnerable to extinction, and we see demographic stochasticity. Here's a skewed sex ratio here, and potential differences in survival between the sexes. The second top model survival estimate was varied by sex, and its potential, there's a difference. And environmental stochasticity, we know that there's a climate change effect. It's changing over time, and it's already extremely harsh environment and higher altitude, and changes can be directly influenced to the population, and they reduce the precipitation limit, vegetation growth, and less food for Gobi bears. And genetic stochasticity, Gobi bears have very low genetic diversity and highly inbred, and potential genetic drift effect, and all this stochasticity interact each other, and it's kind of like can be on in extinction vortex. Second, let's now discuss a second section, and this one describe more about phylogeography, genetic diversity, and connectivity of brown bears in Central Asia, and also we had some samples from other regions. So here we can see brown bears are widely distributed. In previous mitochondrial DNA studies, phylogeographic studies showed 9 to 12 clades, different lineages, and clades and subclades in the world. Basically, maternal lineages show each population regionally clusters into unique haplotypes, and because of those results, brown bears were almost considered to be a model species. When we look at 3A1 bears in the past, we were thinking brown bears in the north are all 3A1 bears in Eurasia throughout, and Gobi and Himalayan bears, one clade, considered to be one clade, and then Tibet and Hokkaido were sister lineages in the past. And then there are Hokkaido have 3 different haplogroup lineages there on maternal DNA. But since mitochondrial DNA only shows one side of evolutionary history, previous data also used only mitochondrial DNA, it's the Harambe control region. We needed to understand the full study. We want to expand this study and ask the following questions. What's the genetic diversity among populations? What's the population structure and how many phylogenetic lineages are present? What's the genetic connectivity, evolutionary history of brown bears, especially in Central Asia? To answer those questions, we have mitochondrial DNA, COX-2 control region, more sequences, and then based after we did whole genome, we were able to take mitochondrial DNA data set, and then almost 13,000 base pair we were able to compare. And for nuclear DNA, 13 microsite loci, 138 pairs in 8 locations. You can see the number of samples from sampling location, and have sequenced 9 new whole genomes from Asia, from this area, and then used previously published 25 pairs. And these are the samples compared. And now there's a new paper also in Northern Eurasia. There are more samples here, but in this map not showing. And we used 9 brown bear, and then 9 brown bears from Europe, and 6 from North America. And we also used other species whole genomes to compare. You get good phylogenetic tree, and grizzly bear used as a genome, used as a reference genome. We did resequencing bioinformatic approach following this, and after all this, filtered out necessary different variant, single nucleotide polymorphism, different 6,800, 4,000 SNPs found, and those, based on those SNPs, estimated the genetic diversity and population structure, calculated all these parameters based on 30 individuals, and for phylogenetic tree, 3 different phylogenetic tree method, and for effective population size used Colston-based analysis. And now we can see the results. For here, we documented high levels of mitochondrial DNA, nuclear DNA diversity in Alтай, Саян, Бүтээлийн Ороо, Hank in Northern Mongolia, but we found very low genetic diversity in Gobi and Himalayan region. The mitochondrial DNA, and then here nuclear DNA microsatellites result. And for the genome-wide diversity observed heterozygosity, here the results showing relatively high observed heterozygosity in Europe and Northern Asia, you can see, but very low in Gobi, Himalaya, Saiyan, Apennine, and mainland North America. Those individuals have very low genetic diversity, and we know that cave bear are extinct, and polar bear, all polar bear are low, and same level as Gobi, Himalaya, and several individuals there are very low there. For the population structure, for principal coordinate analysis based on 13 microsite loci, Gobi were clustered separately in the life axis here in green. And we can also see in the side, if pairwise FSD comparing different population have the genetic distance high, the Gobi are pretty high comparing brown bears in that region. And after that, Himalaya separated. And next one is the Altai bears, different from other bears in the Northern Asia. And brown bears closer to, but interestingly, Gobi bears were closer to Northern and Western Mongolia than bears in Pakistan. And there's a high genetic distance between Gobi and Himalayan macrosatellites. And here we see structure result here, and based on 13 and the basic subdivision K=2 showed Gobi is different from rest of the brown bears. And second, K=3, Himalaya started to separate, and K=4, Altai bears are separating. I can use the pointer. Here we see the result from whole genome results, PCA result here, population structure. We can see Gobi and Himalayan brown bears are distinct from rest of the brown bears. Here we see here each of the color and design showing the different populations, and Gobi and Himalaya are different on this result. And for population structure analysis, here we know that polar bear are different species. It's first separates out in light blue here, and then second Gobi and Himalaya separated, K=3, and then after that the Eurasian bears and North America bear separates here, and then K=5 we see Northern Asia bears separate here, and then after that K=6 Gobi and Himalaya separated, and K=7 we see there's some ancient bears have different. We had a Pleistocene ancient brown bear and also cave bear ones. They are separated on K=7, and here in the structure map is showing K=7 results. Okay, and K=8 show that your differences on in Europe, and but K=7 was the most supported based on the geography and then structure. And what would we learn on your genetic diversity and then divergence, pairwise DXY divergence limit? We see here polar bears are most high distance and high divergence, and after that Gobi and Himalaya most highest genetic more divergent and higher genetic distance. And Gobi and Himalaya have also here very high genetic distance in this whole genome here. And for the now we can look at the phylogenetic tree based on mitochondrial DNA 927 base pair. Here we see different clusters here, and one result here, Gobi and Himalaya in Kumar et al. 2017 already showed Gobi and Himalaya the ancient ancestral brown bear based on mitochondrial DNA here, and it was consistent. And interesting for this result is that we previously expected we would find 3A bears, but we found most bears were 3B bears from those regions. But we also found 3A1 bears in Hankinti mountain range. And when we look at the divergence time estimate based on this data set, Gobi and hemline lineage diverged 690,000 years ago. Cluster B, 3B and 3AB are separated 240,000 years ago. For your maximum likelihood phylogenetic tree on larger mitochondrial DNA data set, the hemline brown bear, Gobi bear separated. They showed monoplatic, showed the splits and called 3A, 6A, and 6B lineages. And for whole genome data set, we found here 5 different clusters, 5 different lineages in Europe, Northern Asia, and then Northern America, and Gobi and Himalaya. Here's is the lineage to the North America, and then they are monoplatic, and we know your polar bears are outgroup distinct, but here we see neighborhood tree also showing similar result here, and okay, now we can look at the discussion. We found large genetic diversity in Apenian, Gobi, Himalayan brown bear population, which is similar to ancient brown bear, cave and polar bears, and bears in Europe, North Asia, and North America had relatively high genetic diversity. This is consistent with the theory that small isolated populations have large genetic diversity compared to large connected populations. And we found 5 potential genetic groups, evolutionary lineages are present in this sample. And Gobi, Himalaya, Northern Mongolia with Russia and Siberia, and European bears, and North America. And based on the effective population size, we can see here in the x-axis, this is the time, 10,000 years ago, 100,000 years ago, 1 million years ago. And here we can see the effective population size, 10,000, 20,000. This number changes over time, and we can see that there were like one main bottleneck here, and then two main bottleneck, and then it's reduced the class glacial. Like 10,000 years ago, they were very low effective population size. The interesting result here is that the European bear, Gobi bear have similar demographic history here, and possibly they had similar good environmental condition to grow this higher during this time, but the Himalayan brown bear and the Northern Asian brown bear, the little increase, but it declined. They were smaller. Probably it's the higher elevation and didn't help. Okay. Now we can look at the results. The mitochondrial DNA, nuclear DNA have a little bit different results, but we know that mitochondrial DNA only shows one side of history, and we know more male bears move longer distance, and they show more of gene flow, and that shows on the phylogeny, but we are also able to see the clusters. There were some similar thing. Gobi and Himalaya. There were some similarities based on comparing those phylogenetic trees. Okay, based on those results, whole genome data set, we see that Asia is the hotspot for phylogenetic diversity, and we know that Gobi, Himalaya, and Northern Mongolia, there are 3 different lineages, and recent publication in it all, the Hokkaido bears were different from the mainland bears, and probably different lineage, and Tibetan brown bears should be studied, will be also interesting. They are different on mitochondrial in a different lineage, and will be interesting this in the future. And future study should be more clear about those questions. And now we can go to taxonomic. What did we learn about taxonomy? So it's very important for conservation. Identifying taxonomic unit or conservation unit is important to conserve the evolutionary potential, unique traits, and biodiversity, mostly the genetic component in this landscape. And to for this taxonomy, there's a different philosophy like based on science, philosophy, or politics, but we know that in general, when there's a higher, more adaptive differences, more separate out, and there can be more subspecies and more separation will show in taxonomic level, but there's a more gene flow, it's more tend to go through populations. And for subspecies, the morphological difference, geographic isolations are main criteria for subspecies criteria, but genetic is the very important criteria to show that show the divergence between the lineages. And it's important to show genomic approach can show that both adaptive and also have neutral loci should be included to show the divergence. And for Gobi, for taxonomic subspecies of brown bears in Central Asia, we can see here for previously Gobi were considered to be like same as Himalayan brown bear, that's based on mitochondrial DNA, short region, but now we see that those bears are monophyletic. They are in criteria for evolutionary significant unit, the Maurice criteria, and Gobi and Himalaya distinct monophyletic groups based on also whole genome SNPs dataset. And morphological, we see there's a morphological difference in different ecosystem in the geographic isolations. Okay, in the future, we need to continue this genetic monitoring, and currently I'm working on this SNPs panel for better individual identification and can find more of their relatives. For macrostylids, since Gobi bears are very hybrid, inbred and low genetic diversity, it's important to find more stronger tools, and its SNPs is the future. And this method should be combined with camera trapping and telemetry dataset. And in the future, more habitat studies should be studied. Oh, sorry. And need to find more fecal samples outside of the sampling area to find more females. And for diet studies, since the bears are living in resource limited area, is understanding more of the ecosystem level of question and diet component is very important, and planning to work on this DNA metabarcode. And one thing is for Gobi bears, they are isolated, small, and so we need to focus on the habitat quality and connectivity. And we found like in the one historical old samples, we found the mixed bear, Gobi and Northern. We found that on macrostylid results, also whole genome data set, we also found shared alleles from Alta and Gobi, So there was historical connectivity, even they are in different phylogenetic lineages. And for future perspectives, augmentation deserves further consideration. It can help with demographic and genetic risk, and also to think about the connectivity between the brown bears in Central Asia. We need to understand this, and should be more studies. Can be landscape genomic, can be a tool to study that. And we also need to identify the adaptive loci for Gobi and other regions to consider the adaptive genetic variations, and most ecologically and genetically similar in terms of adaptive genetic variation should be should translocation efforts be needed, and it's important to maintain the fitness and provide information about the ability for those bears to respond to any selective pressures. Okay. And in the future, we need to continue. This non-invasive sampling can answer a lot of different questions for different wildlife, not only bears. And better conservation management tool is necessary and sustainable long-term study. And in Mongolia, national capacity is important to be improved. And for this purpose, last summer I organized a conservation genetic course, collaborated with professors from University of Idaho, Lizard Waits, and University of Wyoming and Boise State University. And all this scientific information can help with the better conservation efforts in Mongolia. And this can be not only just in Mongolia. We should think about connectivity for brown bears in between countries, because bears don't have border, and good to have more connectivity between those. Try to improve the historical connectivity, increase the genetic diversity. And I would like to thank the people, my collaborators, and people who helped throughout over the years, and funders, and my supervisors. And if you have any questions, let me know. Thank you.
All right, thanks very much, Odka. That's a lot of information. A non-trivial talk. That was pretty cool. You know, I was going to start off with one comment and a question just to get things going. So the Himalayan and the Gobi bear sort of had a common lineage, right? And then they split off, but their FST and everything was the farthest. So the two bears that were the most common split off to they had the FST very, very wide. Is that just because they're small populations, or what, and the genetic drift is really strong?
Yes, it's two of them very small, isolated, and then that must be that genetic drift influence have lower genetic diversity, more specified, left out, fewer specific loci in these regions that showed more of that distance. But if we look at the phylogenetic tree and then divergence, we see they had shared ancestry, but they now split. Yeah, but distance is pretty high. That's like, it must be the recent thing. They are more,
So now to give you a really hard question, after all that, if you were going to augment that population in 10 years, does your data tell us where might be the best source population? And what would that be? I know that's not an easy question. Where would you get bears from?
So, right now, like realistically, we see that there was historical connectivity in the north, in Altai. That's a geographically most closer one to Gobi bears. and like 200 or maybe maximum 300. That can be possible source population, but there are few individuals there. There should be more studies and should do more demographic study. We need to understand how many bears are there, and also it should be expanded through different countries and yeah, figure out the potential options. But
Well, from your talk,
yeah.
that's what I came away with, was that Altai
Mhm.
bears would be the best possible.
Mhm.
I know historically there was bears between there. You know, like in the 1970s, the most recent population of the Gobi was to that northwest, wasn't it? I think so. Anybody else?
I don't have a question. Well, I have a question for you, Michael, or for Bruce. I don't know
That's not fair.
because
I'm sorry. Go ahead.
because I saw a brown bear distribution map, which was created in 2017, if I'm not mistaken. So, could you please show that brown bear distribution map to us? If you can share your screen again.
Yeah.
Thank you so much. I appreciate that. If I'm not mistaken, we are going to update this map, right? Because Hossein Ambrali sent me an email a few months ago, asked for some information. I replied to him, right? I replied to him because this map is out of date. Brown bears definitely occur in two mountain ranges in Iran, in the Alborz Mountains and in the Zagros Mountains. Not this one. Brown bear distribution map. A few slides ago, please. There was a brown bear distribution map. I saw that.
Yeah, it was one, and you're right. Bruce was sort of the lead on that, and I helped him a lot. We both did that map, and with Stefan and Juro Huber. But
Yes, this one.
there you go. So you're
Yeah,
right.
I'm
We're initiating a process, and actually, in another effort, in another Zoom calls, we're just getting an app together where we're going to go around and gather information from many people across that range. So we will be on your case about what you know. So
right, because though this is too small, I had to go like this to see Iran, and in Iran I can see that the north of the country is covered with this historical range. That's blue, if I'm not mistaken. But I'm sure that the current range of brown bears in Iran is still within that historical range. If you see what I mean, that's not a historical range. That's a current range.
yeah, well, there's one of the issues with the map, just the mechanics of getting the map on the website. They actually ended up making a mistake on that.
Right.
Correct.
Right.
It ran. So we tried to fix it. The first map that came out was better, and then they got it screwed up, and I'm not sure what state it's in now. But there's a lot of different. We've been collecting bits of data so far and talking to people. There's a lot more data now to inform this Asian map, and so I think it should be better all across Central Asia.
Okay, I got that. Thank you so much. Anyway,
We're going to be on the case about it, so you're going to have to help. So
the big problem is that the map that we made did not get put on the IUCN site. They missed the Elburz whole mountain range. And it wasn't that, I mean, not that the map is perfect, but it's the main problem was just a technical issue that they never put on the polygon. So one whole mountain range that has bears is not there.
Right, I got that. I just wanted to
Okay.
clarify this, because I saw that, and to me, it was an error, but that's quite all right. Thank you.
Yeah. Anybody else got some questions for Odka?
Mike, there's a question in the.
Oh, okay, good. Yeah, very good. Thank you. I got it open here. Okay, I'm going to read this one from Manu. Females collected less than half as many hairs as males. but it's not conceivable that males with a larger home range, but is it not conceivable that that males with a larger home range collected more than females? In Hokkaido, we also collected more male hairs. We collected male hairs than those of females. However, the sex ratio of the surviving population would be biased towards females. Well, it's just the reverse then here. Anyway, Odka, what you guys went in, and in some of the later years samplings, you tried to change the way you sampled in the hopes of getting more females, but you didn't. Is that correct?
Yes, that's correct. The original hypothesis, yeah, we thought like males are dominating, females not coming to that sampling sites, but we tried to increase that sampling, more sampling sites, and even like during bear capture, there are more males than females. And over the years of this genetic monitoring, we find more males than females.
And I think one of the earlier hypotheses was that males were maybe excluding females from the feeders, right?
Yeah.
And when you look at the telemetry data, it doesn't look like that's happening at all, which is kind of nice. But also, I think you guys put two or three DNA sites near but away from the feeding structure. So hopefully a female who was excluded could get to these other places, but it didn't work out. So it does look a little bit real.
To me, that this was striking the biggest conservation issue there. If the data is real, if you've got that so few females, there's something going on that's interesting academically, but also from a conservation point of view. I mean, obviously we can put out hypotheses of why that would be, and one perhaps is that there are more males, and it's such a generally poor habitat, probably with very local small areas of good food. The males may dominate them, and the females may not be able to access the best food sources. So there's a lot of things that could be going on that are pretty important for the conservation of that population. I think if that that data is sort of frightening. Actually, we have the opposite in most places because we hunt males. We have far more females than males. Our sampling gets lots of males because they have bigger ranges, but the density estimates are higher on females. Where that's if you got way fewer females, that's that's kind of frightening for the population.
Well, I think it's a good hypothesis that the limited food supply is affecting female survival, and they got to grow the kids and wean them, have them suckle for a couple of years, and that I can see it being tougher. And
we also know that the leading natural mortality cause, not human caused in North America anyway, is males killing females. I mean that's their the leading cause of natural death. So there might be something going on there that's pretty interesting and important. But
yeah, that's interesting. I don't think they have any evidence yet, but they're through radio collaring. They're looking. It's so hard to bear dies go out and find it.
Oh well, the sample sizes will be very small. If there's only 15, 20 females, it's going to take you a long time to know using telemetry. What's
we found one mortality that were killed by another male bear. One younger female was killed by older, one large male. That was 2013, and that sample found from that did female SMAV, and that genotype matched with Peru's captured males DNA.
Was she killed by an adult male? Was it an adult male?
Yes, it was adult.
Okay.
Well, there you go. That's kind of interesting. First I've heard of that.
That's very interesting. Yeah.
And I think one of the goals of the telemetry project was to sort out some of this stuff, but they just collar so few bears, it's taking a long time to have those answers appear in the data. We had a bear die the year I was there, the last year I was there. I can remember when Joe Reese was up videoing the bears at Otisingis, I think it was. We watched the bear come in, and then it died right after that. But it wasn't a female, I don't think.
I think that's same one. A female, young female.
That was a female?
Yeah.
Oh, okay. Well,
And what did it die from?
well, I can't remember, maybe Odka knows, but there was nothing apparent.
That same as that I just told, the same case. That case.
Same case.
Yeah.
So Atka,
So-
the 85% survival that you had, that's for what ages? That's just for everything in the hair snares. That's
Yes,
everything,
yeah, all sampling.
everything all
It's-
blended together.
Yeah.
Which is pretty low.
Yeah, that's like a hunted population.
But if it included yearlings and things,
It might be, yeah.
might push it down a bit.
Mhm.
It's
Yeah,
interesting.
during,
First went there, I got there and, everybody said, yeah, we're going to go out and work on bears and catch them and put radio collars on. And I thought, yeah, it'd be interesting to see a bear out here. We drove for 3 days through nothing. And then sure enough, you'd bump into them. So it's an extremely, I don't think until you go there, you really can't get a sense of how unproductive that place is. It's just amazing. It's impressive that the bears are there. So there has to be these bottom up stressors that are really limiting.
I think that's got to be pretty obvious looking at the area.
Ali has a question there, Mike.
Oh, there you go. Yeah, sorry. So,
Yeah,
oh, okay. Yeah, Ollie. Yeah, there's one more in the chat, but let's go with Ollie right now.
okay. Thank you so much. Hello, everyone.
Hello.
And thanks to Abhijit for a very comprehensive presentation. This is definitely an interesting and insightful study covering quite a lot of aspects and enhancing our knowledge on overall the Asian bears. So one, thing I was thinking about this male bias ratio. And I was wondering if there is a practice of cub capturing in Mongolia, because in some areas when the poachers go and try to capture the cub, they kill the mothers. And that may have increased the, female mortality in if that is the case. Because we have seen this practice in some other parts of the world. So this is
Asking, is there any are there any people capturing cubs and killing the mother to get the cubs?
yeah.
How do you think, Odka? I'm not sure there is.
for Gobi, I don't think people are killing Gobi bears because it's considered to be really special animal in Mongolia, and people would. We didn't find. I didn't show any results on hunting results here, but all hunted brown bear population, the illegal hunting samples didn't find any Gobi bear DNA from it. And I don't think people like killing Gobi bears, at least last several decades. But it's possible in the past there may be some bear anthropogenic influence. It's possible, but for now it's very special. Those Gobi bears are very special, and even in government level, people focus on it. And yeah, there are a lot of Gobi bear products, and people truly consider it as a national pride.
So they are protected. So they are protected by both the government and the public, right? This is what you are suggesting.
Yes.
Okay,
It's
that's
called
great.
the park is called a strictly protected area, and you just can't go there. There's like people
Okay.
can't go there. It's very hard.
Okay.
You got to get a permit just to go in. So when you go in there, you spend a month in there and you don't see anybody.
Right.
Yeah,
Great.
only rangers,
Except-
researchers, and people who have permits can enter the strictly protected area.
And
Okay.
this sort of leads into Mano's other question, which is, are there possible anthropogenic or human influences on the bottleneck factors estimated in the demographic history? When you had those bottlenecks going up and down, could that have been caused by people?
That can be like the last 10,000 years ago, or that's possible that it can be human influence. but before that, 120,000 years ago, that bottleneck must be natural thing.
Yeah.
Yeah.
So there was one other element that I was wondering. Regarding the genetic diversity, you're reporting the low diversity in Himalayan bears.
Mhm.
I remember a study in 2017, no, 2007, sorry, a while ago on the Osai brown bears. There we found moderate level of genetic diversity based on different markers of mitochondrial DNA. So I was wondering if you're using different markers, or what is the possible explanation for that?
Mhm. Yeah, this one we used macrosatellites for in also whole genome dataset, and I trust the whole genome dataset. It's showing that genetic diversity, and for macrosatellites, we mostly choose the most polymorphic one for population to identify individual, and each population have specific primers, loci, and that's more polymorphic for that region, and it's very relative estimate, hard to compare overall. If we are using, it has to be like for macrosats, we had to do same lab to trust the results. When we do like different labs, sometimes people call some alleles differently. It's also a possible thing. Another thing is the genetic diversity low, also it showed on the mitochondrial DNA. I think Himalayan brown bears in general have low genetic diversity. We don't have many samples from that region, and maybe like you had more samples that may have helped to have better, higher diversity on macrosatellites, but same time we have to trust the same SNPs used throughout this region and compared, and this can be, both Gobi and Himlai have very low genetic diversity. Have to focus on that part, I think.
Well, I think what was impressive about what you did was, mitochondrial DNA is really just one locus, right? It's just one circular thing. But, you did the microsatellites and then the whole genome. So you sort of tried to cut through markers being a problem here. I think that was pretty good. Well,
So what I understand is genetic diversity you are reporting, it is not just based on the mitochondrial DNA, it is based on the whole genome.
Yes, it's a
Okay.
whole genome. We can check the slide again.
No, yeah, that.
Yeah.
The other thing that I thought was good about your study was that you sampled the other bears nearest to the Gobi, rather than just comparing it to North America and Europe and other parts of Asia. You actually went and sampled around the sort of the umbrella in the northern Russia or southern Russia there, which gives more credence to some of these conclusions.
Mm-hmm. So this is only like control region, haploid diversity. Himlai looks a little higher than Gobi, but it's still, if you look at the other population here, it's pretty low. And when we look at macrosatellites, it's a lower number of individuals. It's possible that this fewer number may influence that, but also same time, it doesn't look like that much of many alleles there, and low allelic richness and low observed heterozygosity expected even on this one. And then, but
Now
it's
I believe these
it's
numbers are
same loci, use same laboratory and compared, not from different studies to compare it. And then this is whole genome, genome-wide observed heterozygosity and very low.
Yeah, but I think your mitochondrial results are comparable to what we published in 2007.
I agree.
Yeah, it was 0.557 you're showing. And what we got, it was probably 0.6 or something like that. And we interpreted that as a moderate one. But in relative terms, if you're comparing with more healthy population, then you're right. It is low on the lower end.
Yeah, like whole genome levels or genomic level of studies, when we use SNPs, compare all populations, like this more clear information than macrosatellites.
Thank you.
Okay, I think I'm gonna turn off the recording.